Good morning, everyone. My name is Loris Pouvreau, and I'm a senior scientist protein technology at Wageningen University and Research. Today I will talk about new source of protein with one main example and also the challenge and opportunities that you can face with structuring of plant protein. This is an overview of the different protein sources that Wageningen University is investigating. One should not discard animal protein, as we believe this circular system, including animal, is more sustainable than a system without animal. Every piece has its own part. When talking about plant protein, we consider three groups: the established sources, such as cereal and grain, the emerging sources with the legumes protein, you can think of soy and pea protein, for example, and the protein from co-products such as potato protein, which are extracted from the potato juice, which is obtained from the starch manufacturing, and protein extracted from green leaf, and I will come back to this example later. The future proteins such as fermentative biomass. You can think of proteins which are extracted in bacteria or yeast, such as mycoprotein, but you can also think of using the whole microbiome biomass without any purification step as a source of protein for food application. The other novel high protein sources can include, for example, duckweed or microalgae, which we believe has a great potential, but it still needs quite some research for being applicable in food. One protein ingredient cannot excel at everything, but we believe that there is an application for all protein ingredients. For example, if you are looking at developing a beverage, you will be looking for an ingredient which is highly soluble, because you don't want this sandy perception in your mouth. On the other hand, if you want to produce a cheese or yogurt, in addition to a relatively high solubility, you will also be looking for an ingredient which is able to form aggregates and a protein network upon heating or acidification. If you want, on the other hand, to develop a plant-based meat analog, let's say per extrusion, a low or intermediate protein solubility is sufficient. You are looking for different properties. You are looking for a certain elasticity, a certain, like in gluten, for example, network, and an interchange of SS bridges. Malfractionation of protein from agricultural side stream has regained a lot of interest in the last 2 or 3 decades. This is an example of mild extraction of the protein from green leaves, and more specifically from sugar beet leaves. Up to now, in the Netherlands, sugar beet are of course cultivated for sugar production. Within this production, you are left with 40 tons per hectare of green leaves. The challenge that you face if you want to extract protein from green leaves is that this extraction has to take place on a very short term, otherwise a decay will take place of the leaves. Many steps, and I will not go in details, but many steps are required to go from the sugar beet leaves to the protein ingredients, but I will mention a couple of them. You of course need the macerations to be able to make a juice out of the sugar beet leaves. You need a solid liquid separation for separating the soluble and insoluble material, and you also need to filtration to concentrate your stream, a color removal step to be sure that you don't have any green color or yellow color left, and a spray drying step, of course. Within these extractions, you also are creating some co-products which can also be valorized. Some of it can still be used because it still contains quite some protein as a feed in feed. Otherwise, the other stream can be used for biopolymer production or for biogas production. One may wonder why Rubisco. Rubisco is a very highly conserved enzyme which catalyze the first major step of the carbon fixation in green leaves. It is found in all green leaves, and therefore it is actually the most abundant protein in the world. And in addition, which is also very interesting for its functionality, it is water soluble. Rubisco is a very interesting protein source for application, as it has a good nutritional value based on their amino acid composition, but also a relatively good digestibility. It has extremely good functional properties, and I will come back to the gelling properties in my next slide. Moreover, it's also very low allergenic. So coming back to the gelling properties, I'm showing here two examples. On the left, an example of sugar beet protein extracted from sugar beet leaves, and on the right, protein extracted from spinach. Why am I comparing these two? It's just to show you that the results are actually quite similar independently of the source of the leaves they are extracted from. So if we look more precisely on the left side of the screen, on the sugar protein extracted from sugar beet leaves, you can clearly see that Rubisco is able to form a gel at a much lower protein content than soy or whey protein. This is also confirmed with the protein extracted from spinach, where clearly it is even gelling at lower protein content, but also forming a stronger gel than egg white protein. For that matter, it's also, we think it has great potential as a dairy alternative, so to be able to form as one ingredient of a plant-based yogurt or plant-based cheese, or also great application as an egg replacer, as a binder, for example, in plant-based meat analog. The second part of my talk would be on processing protein towards plant-based food, and more specifically plant-based meat analog. What we are really paying attention and working on at the university is on the quality of the plant source material. So what are the properties of the protein in the fraction? What purity is required for obtaining a good final product quality, and also try to make, to use less refined ingredient in general. What we also pay quite a lot of attention is how these different ingredients, so protein, fiber, oil, or flavor, how do they interact with each other during the processing? And by processing, you can think of extrusion, for example. One thing which should not be neglected is post-processing steps, which are often quite required after the extrusion process, for example, which are marination, freezing, thawing, packaging, and shelf life, of course. Last but not least, what is also very important and what is the main driver for consumers to buy a plant-based meat analog more than one other one is the texture, so the bite during chewing, and the juiciness, which that people are expecting to be closer to meat. If you look at the meat analog market, you can clearly see that the plant-based meat analog is very much dominated by two ingredients, soybean and wheat gluten, which is for good reason. They are quite functional in this type of application. What is very important is to switch, and you can see it already happening to other type of proteins such as yellow pea or lupin, for example. If you look at the technology available for texturizing protein towards meat analog, there is different of them, different possibility: mixing, forming, and combining of ingredients, fermentation, like a corn product, for example, with a mycoprotein. But I will mainly focus my talk on the extrusion, the shear cell technology, and the 3D footprinting. When talking about extrusion, you can see a new trend towards high moisture extrusion for plant-based meat analog. These offer more fiber structure which are mimicking the structure of meat, or closer to what the consumer is expecting, and may also offer more flexibility in type of ingredient to be used. During the high moisture extrusion, of course, there is some important step outside the selection of the ingredient. You can think of the extrusion conditions, so the temperature profile, the shear applied during the cooking step, and the pressure applied, but also the importance of the cooling die, which fixates the structure. Of course, one should not neglect that post-processing steps are taking place. So in some cases you have a freezing and thawing cycle to actually get a clear layered structure, and a marination to bring some additional flavor to the extruded material. It is quite widely used in industry, but as I said, it regained interest in the last 10 years. What is our interest is to use other protein outside soy and gluten, or to use combination of different type of protein. What we also look towards is understanding the extrusion of what is needed in the protein side to actually be able to form this anisotropic structure, or this fiber structure which resembles the one of meat. And another aspect that we are also working on is product quality, so in terms of texture and juiciness. The second technology, which is relatively more recent and which is not industrially available yet, is the shear cell technology. On the left, you can see a picture of the conical shear cell, and on the right, you can see a schematic representation of this conical shear cell. The advantage of the conical shear cell is that you apply a very simple shear, and you can see it in the drawing, or at least I hope you can see it, is that one of the cones is fixed and the other one is rotating, so applying a specific shear, but a linear shear. The same principle applies to the quad shear cell, where the shape is different, but the principle is the same, is one cylinder is fixed and the other one is turning. As you can see in the bottom right, you are also clearly getting a very nice fiber structure, so not as layered as by extrusion. So you get different product properties using the shear cell when compared to the extrusion. A comparison between extrusion and shear cell, both of them are able to form anisotropic structure or fiber structure, except that quite often the extrusion gets more a layered structure when compared to the shear cell. The similar basic step, mixing and hydration, thermomechanical treatment in both cases, and of course in both cases you have a cooling step to fixate the structure. Similar formulation in terms of protein can be used for both technology. The main differences are on the fact that the shear cell is a batch process, while the extrusion is a continuous process, and as I said, the shear cell is not commercially available yet. The window of temperature, higher temperature are used in extrusion, but shorter residence time, in the shear cell you have a simple shear flow, while during extrusion you have a simple shear flow plus an elongational flow. 3D printing for plant-based food or plant-based meat analog has gained a lot of interest in the last five years, with the fact that 3D printing offers the flexibility in shape, in using personalized recipe, in creating different structure. The different structure, for example, can be obtained by having a biphasic system with different texture, or by coaxial printing to be able to have a different core and shell material. We believe that it offers quite a lot of opportunity for plant protein in terms of improving the texture and the juiciness, but also in combination of protein and the ability to form different layer structure. This brings me to my take-home message. Protein transition toward more plant protein, we believe, is necessary from a sustainability point of view, but also to be able to feed the world in the future. There is a need for broader range of plant protein suitable for different types of application. Again, a need for an integral approach for isolation, fractionation, and structuring, less refined ingredients for a better use of our crops. And last but not least, there is a need for a better understanding of plant protein functionality in application. And with this, I would like to thank you. And if you have any questions, please don't hesitate to reach out.
Yeah, good evening, everybody. My name is Carsten Krom, and I'll be talking to you today about opportunities in the protein space. I will come more from maybe a commercial perspective than my colleagues. I'll talk a little bit about my background first. So I have a PhD in fish nutrition from the Institute of Aquaculture in Stirling, and have since worked in investment, and this in particular early stage venture capital investment in the field of aquaculture. I've done this in since 2014, first on payroll for a fund, and since 2016 sort of on my own first for a year, and then co-founded Hedge together with Wayne Murphy and Georg Baunach. Hedge is an entity that is making investments in early stage aquaculture technologies, including a lot of protein technologies. We have offices in Norway, in Hawaii, and in Singapore, with the intent of providing value to the investments that we make, in a sense that we connect them across those continents to potential customers and partners. So we've got 13 people in total in the team. We also do a lot of consulting. So we consult corporates on their innovation strategy when it comes to among other things, protein and their investment strategy. We consult them on due diligence. For example, we just finished for a large Japanese multinational a due diligence on insects, and we also, and this kind of stuff is what we do. So given I have a background as a fish nutritionist, I tend to think that I can still talk about the technical side of things, although I've also noticed over the last years that if you don't stay in science, you kind of lose quite a bit of those skills, but to an extent, I still know what an amino acid is and a peptide, so I can still be dangerous. But right now the focus, or my professional focus, has been around making investments, and today's talk will be around how those investments in protein can look like and what type of opportunities we see in this space. Yeah, with this, I'd like to already enter. So we, on our end, define 5 different fields, which you see here. Just generally speaking, you can produce proteins, obviously, for the food market, but you can also produce them for the feed market. And again, generally speaking, any particular protein can, in most cases, be sold to either markets. It just becomes a function of price and production cost at the end of the day, which markets you target. There are some proteins which can be used as feed, but not as food for various reasons. One is, for example, that humans may have allergic reactions against particular proteins that animals don't have. There's 2 or 3 examples where this is the case. And others might just have to do with regulatory issues. So for example, if you have insect proteins that are where the larvae are fed on essentially excrements, on waste coming from toilets, you're not allowed in most regions to feed the products there directly to humans, but you may feed them to animals. So, but from a structural perspective, from a nutritional perspective, there's no difference between proteins sold into the food and proteins sold into the feed market technically. So in terms of the 5 main subsectors for protein that we've identified as the high potential subsectors, you will see here microalgae, seaweed, insects, single cell proteins, and new plant proteins. I should say that there's very fundamental differences between those, and we have a very distinct or differentiated view on what we consider to be the commodity proteins of the future, essentially. And I'm happy to just briefly go into detail here. So microalgae has already had a bit of a boom in the noughties when the producers were looking at microalgae as a replacement for biofuel. I would say it has a bit of a renaissance now because microalgae produce omega-3 fatty acids, the long chain omega-3 fatty acids that you only find in fish oil usually. Microalgae produces those. And so they are more and more now produced these days as a fish oil replacement for feeds, but they're also produced as a human nutraceutical, omega-3 nutraceutical. But more and more it becomes possible also to potentially target the protein market. So growing protein-rich algae for the food market or even potentially feed market. There's very different production technologies within microalgae. So you would have the photoautotrophic microalgae that need sunlight and do photosynthesis. You would have the heterotrophic microalgae that consumes organic material like sugar, or in some cases also acetic acids, or other type of organic acids. And it's been traditionally shown that and they grow in different systems. So heterotrophic algae would grow in fermenters, and photoautotrophic algae you can grow in photobioreactors or ponds or greenhouse type of structures. Experience shows that, or what we've seen so far, that fermentation is the more cost-effective way to go. So this is counterintuitive in a sense that you would think if your raw material is just light and CO2 for photosynthesis, this is cheaper than if you have to put sugar in a tank. But there's various reasons why you would actually have it more effective if you grow algae in a tank from a cost perspective compared to, for example, photobioreactors, which cost a lot of money to build, and then, at least as of today, still have a couple of issues when it comes to running them cost effectively. The second subsector, seaweed, is an interesting one because it's very hyped at this stage. There's also here very big differences between the types of seaweed. So when you look at the kelps, they actually have hardly any protein. They mainly have fibers, because they, as you can probably tell, they have a structural component, much like trees. They have to stand upright in the water, and this is the fiber. And fiber isn't necessarily zero protein in it. There are some types of seaweed, like ulva, for example, which has a higher amount of protein, which could at some point become relevant for the food sector or for the feed sector. However, I think that the seaweed as a feed source, and I'm not saying there's not other things that you can use seaweed for, because I think you can, and I actually believe in the sector that I think you can extract very, very valuable microcomponents from it, and that there should be a lot more done, and there is being more done to kind of try to commercialize specific parts of seaweed or specific nutrients that exist in seaweed. But as a bulk protein source, I don't think the kelps are going to be relevant. But there may be some particular seaweeds that may make it at some point. Another big hype is the insect sector. So here we have the black soldier fly, which is the most produced insect at this stage. And then you will also find mealworm, Tenedebrio, molitor, and also crickets in some case. And then I'm sure there's some other species that are also grown commercially. Now, I will say that globally, at this stage, there's only about 10,000 tons of seaweed a year grown, of black soldier fly insects per year grown. And this is nothing compared to the fact that the sector has been already around for a good 6, 7, 8 years. The reasons, I suppose, is that it's just not easy to scale any factory, but in particular, probably factories that look like insect factories where you have to think about more different parts than for other factories. You need to have a very consistent raw material / waste stream at the beginning. So you need to basically put equipment in place to make this a very coherent and permanent or consistent raw material stream. Then you have to have the growing units where the larvae actually eat up and stuff themselves to become, to become big. But then you also need to take a certain fraction of these larvae and hatch them into flies to then replicate and make more larvae, so you can begin the process again. And you obviously have a lot of processing downstream with whatever you do with the dried larvae as you harvest them. And so that's a very interesting sector, but it's also, well, it's not lived up to its promise so far, and we are quite skeptical also as to whether it will do that. Now, I will say this counts for feed. So mainly when we think as aquaculture investors, we think mainly about the use of feed. We don't think as much about the use of food. The food sector for insects, it's a consumer question. I think that's where insects really would have a chance. And I personally would also, wouldn't mind having insects in my daily diet. So I think this is maybe where we could have some consumer shift and thereby help this very sustainable sector to its get it on its feet. Then two also very relevant sectors. I start with single cell proteins. So single cell proteins are essentially bacteria, and they are those bacteria that you can grow in bulk on different raw materials. So again, there's very different types of bacteria or single cell proteins, very different raw materials. So you would have, for example, you can have sugars, you can have certain waste streams, you can have methane, you can have methanol, and very dependent on what type of raw material you have very different economics of those types of projects. And I personally think one beneficial thing about single cell proteins is that they can actually have 70+% protein in the end product, which is quite relevant when it comes to producing protein concentrates and whether this is for the food or feed market. But you would find that the natural protein content of your bacteria is higher than, for example, of your insects or your seaweed or even also your microalgae. And then lastly, new plant proteins. This is something that is, you could see. So for example, in aquaculture, you would have had fish meal as the main protein ingredient all the way up to, I would say, 2008, 2009, maybe a bit earlier than that. But since that point in time, soy protein has sort of replaced large parts of the fish meal. There's still a significant amount of fish meal being used, but soy protein is the other kind of standard ingredient in the field. And so you when you talk about new plant proteins, I do not necessarily think that soy would be considered a new plant protein, but there are other way more sustainable plant proteins that we've been working on, or we've made one investment in particular into a company producing barley protein, barley being a much more sustainable crop than soy, in the sense that it can be grown in temperate regions. It can be grown local to where the feed markets are for salmon, like for example in Canada. Yeah, so with these plant proteins, I think we need to be careful on what we define as new proteins and which ones have already been around for a couple of decades, but there's certainly a lot of potential in those as well. Okay, where are the opportunities in particular as we see it? And maybe here it would be also good to just briefly explain how we think about the protein market, again, from a broader perspective. So if you look at, for example, alternative seafood or alternative meats, and by that I mean cell-based seafood and plant-based seafood, we consider plant-based seafood as potentially very interesting because the raw materials for the plant-based seafood sector are already there. So when you make a plant-based, and that counts for plant-based meat as well, plant-based product, you can use the soy proteins of this world, but you can also use the new ingredients like barley protein. But you've got existing supply chains already in place, and you can it's all about processing these ingredients in a way that give you the best taste. And that is really what it all comes down to. And for this reason, we consider processing technologies as being a very high potential sector in the next couple of years, because we think that also from a price perspective, the plant-based seafood sector has a chance to tackle the existing conventional seafood products. And also flavoring and texturizing will make massive difference here. So if there's lots of, actually, or quite a few plant-based seafood products already in the supermarkets, but frankly, only one or two that we've tried, really you would not be able to taste the difference between real seafood and this plant-based seafood. And so, but we expect there's a lot of potential for improvement here, and this will mainly come down to finding new processing technologies, finding new texturizing technologies, and also if you can, put some kind of flavoring in there that represents the taste of seafood, that would make things easier. So it's talking about plant-based. There's also the cell-based sector. Here, you're obviously relying on a whole supply chain to establish itself from scratch. So while some companies can maybe produce very, very small amounts of lab-based meat or seafood, you do want to look at the whole supply chain, and you need new engineering solutions. You need new scaffolding for the is apparently a big issue to find the right texture. So you need to invest in technologies that produce the scaffold of the seafood or meat. It's not so important if you've just got a burger patty, but if you want to have a whole piece of meat, then obviously you need that. And the growth media in which those cells can grow. Right now, the cells grow in a bovine serum, and that is obviously completely unsustainable and way too expensive. So finding new, more competitive means of replacing this media to grow cells in is the way to go. Nonetheless, as a venture capital investor, we would not rule investments out in this space, but we are also conscious of the fact that rarely you rarely find one company being able to solve all these issues. So if you take a bet in a particular company with a particular technology solving a particular issue in the supply chain, you still need other issues to be fixed where you have no influence in before your investment will provide a good return. And that is sometimes a bit overly risky. Nonetheless, really, really interesting sector, and obviously really booming at this stage as well. Okay, and just another point I'd like to highlight is the use of byproducts, because in many, many cases, the use of byproducts makes the difference in the in whether a technology is economical or not. So if you, for example, take insects, you've got 3 different products. You've got if they refine it or if they process it to those 3 different products, you would have insect oil, you would have insect protein meal, and you would have what's called the frass. And by volume, the insect protein meal, which everybody talks about, would actually be almost the smallest part. I think the oil might be smaller. Yeah, the oil is still a little smaller, but the by volume, the majority would come from the frass. So whether you can sell the frass for 100 or 200 US dollars per ton makes a massive difference. And finding use cases for byproducts, and this actually counts for whether it's single cell protein or most new protein sources have byproducts in their production processes, and in most cases the value of those has a very, very high impact on the economics of any particular production process. So finding use cases for the byproduct is essential. Okay. Yeah, and with this, I think I'll stop here, and I'm happy to take questions or anything like that, but no. Thank you very much. If you have questions, my email is carsten@hedge.blue. Just email me, and thanks for everything.
Good morning, everyone. So I'm Anne-Marie Germick. I am a research associate in Phyllis Coxell team. So Phyllis is an assistant professor in the Department of Food and Human Nutritional Sciences at the University of Manitoba. So the project I will talk about today that we are doing at the U of M is financed by the Good Food Institute. And so I will talk about ultrasound as a tool to monitor plant-based meat analogs during processing. So in fact, you have many actors in these projects. So people from the university in the food science department, like Dr. James House, but also in the physics and astronomy department, Dr. John Page and Dr. Anatoly Stribulevich. And we have also people involved outside of the university, so Dr. Mehmed Ulbek from AGT Foods and Ingredients. So a bit of background before I start about the project for real. So we have a growing demand currently from consumers to get plant-based food in order to to vary their diet or to try new products. So also transformation of plant proteins is a very good way to add value to prairie crops. Wheat, canola meal, soybean, pea, oats, all of those can be transformed to make really products that have better value than the raw product. So one product that gains interest are plant-based meat analogs, which is a product based on plant proteins. It can be any plant proteins, like all the stuff that we are growing in Manitoba, for example. And this meat analog, in fact, is mimicking the fibrous structure of meat. You can see, for example, here, 2 meat analogs, so like a chicken-like material, and the Impossible Burger. So we are still facing challenges, though, to make plant-based meat analog perfect. First, the sensory properties of meat analogs are far from perfect. We are unable, for example, to mimic chewiness and juiciness of animal meats. Also, we have some concern about nutritional properties because, like meat analogs, have less proteins, and the quality of the protein is also not as good as in meat. So the question we have is which processing condition will ideally create the desired sensory and nutritional quality attributes of plant-based meats? And how do processing condition, food formulation, and the mechanical properties of foods will interact during manufacturing in real time? So in fact, monitoring the texture in real time is what we want to do. It will be an asset to understand those questions. So here is a big picture of the project. So we want to produce plant-based meat analog, and we want to, in fact, understand what is making a good sensory quality and what is making a good nutritional quality. And in fact, what is influencing those two things are, in fact, the processing parameters, so the way you are making the product, and the ingredients, what you put inside, like the type of protein and the amount of water. The problem is that you have strong connections between those things, and it's difficult to separate them. What we want to do with the ultrasonic technique is to be able to connect the sensory quality and nutritional quality to those two basic things, processing parameters and ingredients. And basically, looking at the ultrasonic signal will be enough then to know if the product is good or not in real time. So that's really the end goal that we want to achieve, is to really look at the ultrasonic signal to determine if you have what you want at the end. So before talking about ultrasound, I will talk about meat analog processing, how the extrudate is made. So currently, the most common way to produce plant-based meat is extrusion. And so you put plant proteins, you put water inside a big barrel. In the barrel, you have counter-rotating screws. And so the product will be mixed, then sheared, then kneaded, and pushed through a long cooling die. So inside the barrel, the product is cooked, and in the long cooling die, the product is cooled down, and the protein will align to form fibers and a product that look like, for example, chicken meat like here. So the end product is called in the presentation extrudate. So our research plan is to investigate, in fact, the effect of screw speed, moisture, and also the temperature in the barrel, which are the different things that we can play with. So what's happening in the barrel is, in fact, a very complicated process. You start with basically plant proteins, you put water, and what you obtain at the end is something that is structured like that. So a lot of things are happening. So you blend, you hydrate, you mix a new shear, you heat your material, you have some pressure buildup. All of those, in fact, will denature the protein and break some bonds and change a lot of things inside. And then the protein will align inside the cooling die and will make this fiber structure. So now what about the technique we want to use? So we want to use ultrasound to measure the mechanical properties of our material non-destructively. So by mechanical properties, I mean like hardness, softness, squishiness, like or how the material can be extended, this sort of thing. Some of them can be picked up with ultrasound, like kind of. So that's really the general idea. So the example of previous research that were done is, for example, like offline, you can detect bubbles and monitor their evolution inside bread dough. So bread dough is constituted of 80% of bubbles, but you cannot see them. It's completely opaque. And so ultrasound can be an efficient technique to really assess and see the bubbles and monitor how they evolve in time. So that's very good for opaque materials. Online, another study was done in noodle dough and during noodle dough productions. And we were able to monitor differences in composition during the production, like a change in the amount of salt or a change in the amount of water. And also, it was possible to pick up thickness fluctuation, like if the dough is thinning during production, or on the contrary, if you have folds of the dough during production. So it's a very good non-contact quality control tool that can be used on the production line, and non-contact means also that you have no contamination, that everything is done without touching it. So ultrasound can really benefit the food industry. So here we adapted this ultrasonic setup on an extruder to measure ultrasonic properties of our material during production. So we use, in fact, air-coupled transducers. So it's an emitter and a receiver of sound. So you emit a signal, it goes through the extrudate, and you pick it up at the end. This technique is non-destructive, non-contact, fast, not expensive, and very effective in opaque material. So ultrasounds are in fact just high frequency sound waves. So you cannot hear them, but they have the same nature as a regular sound. We are using here low intensity ultrasound. So high intensity ultrasound is sometimes used in processing to modify the properties of material, but those ultrasounds are high intensity. In our case, we want to use low intensity because we don't want to modify the properties of our material. We are using ultrasound only as a probe. So what we measure is the velocity and attenuation of ultrasounds, and they depend on the material where it propagates. So the attenuation coefficient means how much of the sound is lost due to scattering and absorption. And the ultrasonic velocity is how fast the sound wave travels inside the melt. So the two things that we can measure when we do a measurement. So if you have bubbles, for example, in your meat analog or in any material, or if you have layers, it will affect the attenuation and the velocity because sound will scatter on the bubble and basically be sent in all directions. So you will lose some sound. The velocity and attenuation also depend on the material density and the mechanical properties. All those things are connected together. So we are using a setup that is allowing us to measure that. So the setup, I called it the Extrusion setup, like extrusion and ultrasound. And it's placed basically at the end of the extruder barrel. The material will flow in between the transducers on an inclined plane which has a slit. And the inclined plane is here to guide the material in between the transducers. So we have in fact our emitter and our receiver, and the way we are doing an experiment is that first we will acquire a signal in air, and this signal will be basically our reference. It will be the maximum intensity signal that we have, and then we will compare it with a signal that goes through the material we want to study. So this is here with a sample, and depending if the material is hard or soft, you will have different signals. So the general idea is to compare the signals through air and the signals through the material to measure the velocity in the material and how much the ultrasonic wave is attenuated. So for example, here is an example of sort of samples that we got. So we have here like very tiny fibers and everywhere, and those fibers were very well aligned in the entire sample in the same direction. Those samples appear to be very light. When you put them into water, they float, and this is a strong signature that they have bubbles inside. And bubbles, I told you before, means low velocity and high attenuation, and that's what we observed when we did the measurement. We have here a low phase velocity and a high attenuation with those samples that presented some texture. For this experimental run, we had other samples which were not texturized, and those ones didn't have any bubbles inside. So, however, like you can have probably texturized material with no bubble inside. So it's not a systematic connection. It's not because you have bubbles that you have texture, and it's not, the opposite is not true either. So depending on the processing condition, soft material, you can have soft material or harder material, you can have meat-like fiber texture or not, and you can have small bubbles or not. And all those things will give you different ultrasonic signature. And here is a very clear example of what I mean by that. So ultrasonic signal is presented here, and it's after some processing. So basically it's the envelope of the signal. We will compare here two different extrudates. The first one is the extrudate I just showed you before, it with these small little fibers. And it had a very tiny signal compared to the other one, which is another extrudate, which presented like fibers also, like a texture, but more layered. And it was, you see that the fibers were not aligned completely in the same direction. This material also was much harder than the one below. it to the touch. Yeah? But otherwise, if you don't touch it, if you look like that, they looked very similar. So this material was harder, didn't have any bubbles inside, and we can see that we have a very high signal compared to the red one. We can see also another thing is that the signal is arriving earlier than the red one. It means that the velocity is much higher here than here, where it's much lower. So you can see here that even though the material from the outside look similar, when you look at the ultrasonic waves, you can see very different type of signals. So in a way, it's a way of assessing what you are seeing without touching your material. It's a way of doing things, looking inside, while you, with your eyes, you can't look inside. So as a conclusion, low intensity ultrasound is a good potential candidate for food quality control because it's not destructive and it's cost effective and non-contact. And changes in material property induce changes in the ultrasonic signal. And now we are continuing, of course, our research. We want to correlate those processing condition, textural properties, and ultrasonic properties at the same time for different parameters. And so, like, we want to do a full map of what we can see. The future work will introduce a new set of transducers that are in contact with the dye to monitor like how the fibers are formed. And we want to do really systematic texture measurements. We want also to measure the protein quality of the extrudate. And after all of that is done, we want to model the relationship between ultrasound, texture, nutrition, and implement the new knowledge to monitor the extrudate quality in real time. So the end goal will be to scale up the ultrasonic system and maybe apply this to other food applications. So before finishing, I would like to thank the different partners and especially the Good Food Institute for the financing and all those people that are involved in the project with their technical expertise, either in extrusion or ultrasound or like design and manufacture. So, and I would like to thank you also for your attention.